Okay, let's start today's lecture. So if you can hear me as usual, you can raise your hands or chat. Okay, that's great. So at least it's working. So we are continuing to discuss the session 3 and 4, which is the population structure. And okay, let me pull up the slides. We're going to cover the population structure and the inbreeding coefficients if the time permits this week. Then that will be the 2 topics this week. And then we're going to cover the review session on Monday and the exam will be Wednesday. But I mean, if we're not finishing the topics this week, then the schedule might be delayed. So let's see how it goes this week. And the population structure, we're going to cover some key concepts related to population genetics and also understanding the modern genetics as well. And I put the famous quote from the David Wright from his recent book. David Wright is one of the leading experts in the population genetics in our days. And if you are if you have the burning desire in population genetics and how genetics is perceived in the sort of the large scale the era, I highly recommend his book. Probably the book title is The Mixture, and it's also the translated version appeared in the bookstores, so you can easily access to his book. He's one of the also the deep experts in population genetics, and also talking about the human origins as well. So I can guarantee that his book is quite insightful and tell you much about the latest population genetics theme. So if you find interesting over the session 3, his book is yours. Okay, so what we are going to learn today is we're going to introduce the sort of the basic factors about population diversity and evolution. And also we are discussing about the models of the population structure and also how we estimate population structure. And also, as usual, some learning materials. Okay, so the first thing is population structure. So we cover the 3 topics in this section. So first one is just the overview of the population structure and the concept of the allele frequency, which is repeated things as previous topic. And the last one is the 1,000 Genome Project, which is the first evidence to look at the distribution of the allele frequency in a large-scale genomic project. Okay, so what is the population structure? So population structure refers to the presence of systematic difference in allele frequency between subpopulations in a population. So if you look at the genetic variation within and among human population, this can represent the origin and long demographic history of humans. The reason is that the allele frequency is quite different between subpopulations, and that is fully represent, which allele frequencies belong to which population. And population genetics is a field to study genetic difference in natural population and how evolutionary process shape our genetic difference through the time. So genetic difference in this context means the allele frequencies in an organism. So one way we can understand this population structure is then the out of African theory. So when we look at the human history, the first origins of our human beings is from the root from the Africa, right? So some of the African population move out from the Africa and migrate to other places, and that is the early history of the human species, and that is the sort of the source of why we have all different the population structure in our species. And according to out of Africa theory, a small set of the population migrate out of the Africa in the past about 100,000 years ago, and they rapidly expanded throughout the broad geographical region. So some of the people migrate to East Asia or Mid East or Europe or Australia or other, the American continent, and they set in there, and they undergoes their own migration history and also the settlement, the time, and they shape their own genetic background in their region. So non-Africa population has different variant frequency or allele frequency due to the genetic bottleneck or the long migration history. So if you compare non-African population to African population, so first distinction is that we have the different starting point of the population size. So non-African population has the small the population size due to the small sets of people migrate from the African continent. So if you look at the African population at the ancestral time, they have much larger population size, which means that they have much deeper genetic backgrounds than the non-African populations, and it's also reflecting in the allele frequency as well. So if you're talking about the allele frequency, we can say that the allele frequency are quite different by subpopulations, and also this allele frequency has been dynamically changed over the time as part of the evolutionary process. So if you think about the allele frequency in an Eastern Asian and allele frequency in European, they of course have the different frequency as to the certain allele, because they are having the different times and also different evolutionary process, and also the migration history as well. So they have the different composition of certain alleles, and that is represented as a numbers, which is the frequency in each allele. And if you look at the certain allele, then we can see the differences in terms of the allele frequency across the population. So if you look at here, we have three different SNPs here, and three different populations as well. And if you look at their frequency across the populations, they all have the different the combinations of the frequency across different SNPs. So when we think about the population structure, the easiest way to look at these differences or stratification is looking at the allele frequency, and also of course using the latest genomic technology, we can easily measure the allele frequency across the population, and we can infer the population structure. So one of the earliest way to look at this the population structure and also the genomic technology was by the project named 1000 Genome Project. So 1000 Genome Project is the large-scale genomic research that provides an allele frequency in a population. And 1000 Genome Project started at 2008, so which is just after the HandMap project, and using the same experimental design we used in the HandMap project, and they move on to the different topics, which is looking at the distribution of the allele frequency across various populations. So this case, they don't use the microarray technology, which is used in the HandMap project, but now they use the next generation sequencing, because people at the time were able to look at the access to the latest technology at the time, which is the genome sequencing technology, looking at the one base pair resolution rather than the microarray type. So they're looking at all of the genotypes using the direct approach, which we discussed last time, and they access to all of the nucleotides in our genome. But at the time, they're looking at the 4 times depth, which is appeared in here. The reason is that, I mean, nowadays, if you look at the nucleotides by the next generation sequencing, you normally use like a 30 depth or 100 depth, but at the time, the price and the cost was matter. So people only look at 4 depth, which is looking at 4 times at a 1 locus rather than 30 times or 100 times. So a bit of lower confidence than these days, but given the time we used at the next generation sequencing in 2008, that was sort of the high tech approach. So I mean, that was sort of the best solution at the time. And 1,000 genome projects using these 4 times depth next generation sequencing, they record all of the genotypes from the individuals. So this is way different approach than the HEMMET project microarray approach. The reason is that we literally look at all of the nucleotides in an individuals, then compare their genotypes to each other, and we can much detailed information about the population structure and also the allele frequency differences. Okay, and as the HEMMET project, 1,000 Genome Project has the several phases to update their genomic resources, right? So they're not just looking at one time, they're looking at the first phase, second phase, and third phase, then they update their research and also discuss their findings with other peers in the scientific communities, right? Okay, so what they've done in the 1,000 Genome Project. So first thing is they looking at the different ranges of allele frequency in our genome. So what they've done at the first time was to define the frequency group by alleles, right? So they define the variants which are commonly in our population. So we call the common variant. And then we also define the rare variant, which is rarely present in our population, literally. And the low frequency variant, which is the frequency range between the rare variants and common variants. So common variant is the allele frequency greater than 5%. And this is well represent the LD structure in our genome. And this is also typically contained in a SNP microarray, right? So microarray also use the common variants because the common variants working as a text SNP and representing the LD structure in an individual or the population. Then the rare variants is the types of the variants only can identify by next generation sequencing, because the direct approach can read all the nucleotides in our genome. So these things can be done by microarray. So because this is uniquely present in an individual, so not really commonly present in a population. So this is the sort of the merits of using the next generation sequencing technology in 1,000 Genome Project, and they define this rare range, which is less than 0.5% allele frequency, and within that range they define the rare variants. And then the variance between the rare variants and common variants, which is the 0.5 to 5% allele frequency, the 1,000 Genome Project called them low frequency variants. So these are not working as a text SNPs or represent the LD structure, but also somehow the commonly present in our population. So they put it into the different group, which is the low frequency variants, right? So one thing to note that is using this threshold, the 1,000 Genome Project used this way, but this definition can be changed by studies. The reason is that the size of the project, size means the number of individuals participate in a project, is differ by studies. And also, you can surely imagine that the latest project has much larger sample size than that day. So we can definitely looking at much deeper and much rarer variants than defined in 1,000 Genome Project, right? And also we can more precisely, bring the interpretation as the rare variants like less than 0.1% is really rarely present in our population, right? Or sometimes these days people also define the group like ultra rare variants, which is then less than certain percentage. So there is also see the distinction from the rare variants, right? So ultra rare variants is literally much rarer than the rare variants, and they distinct that the characteristics of the ultra rare variants compared to rare variants, right? And also people also define different the types, which is the private variants, which is the only seen one individuals in the study, right? So this is much rarer variants than rare variants or ultra rare variants. Why we care about the private variants? The reason is that if you look at the 10,000 individuals or 100,000 individuals or a million individuals, and if you find private variants which is only seen one time in a single individual in the study, there are likely to be large effect variants compared to other types of variants, or that private variants could be de novo mutation, right? So, I mean, most of the, the population experiments, we don't have the parents' information, and we have no way to deduce the whether this variant is the de novo or not, because we don't have the family information. So in that case, the private variance is the only way we can estimate that variance is appeared as a de novo in a generation or not. So private variance working in that way too. Okay, so back to the 1,000 Genome Project research. So they definitely take an advantage of looking at the rare variants in our population. And what they found that the rare variants, as expected, they are much more likely to be population specific. The reason is that if you look at the common variants expectedly, shared by large proportion across the population. So here is looking at the Central European CEU and the European African YRI and the Han Chinese and Japanese, which is representing the East Asian, and looking at the three population, 85% of the common variants are shared by them, right? But the rare variants is surprisingly only 2% shared by the populations. The reason is that the rare variants is appeared in a near generation. So if you think about the coalescent lineage, so if you look at the common variants, then that variant probably inherit from the very old generation, right? So probably before the time of the divergence. So when two populations diverge out to different, the populations, they has to sitting in the founder population. So that's why they can be commonly present in across the population. So if you think about the common variants, that could be the variant already present in a in ancestral African, or the variants probably in the very early days of the out of Africa migration time. So way before the divergence. So that's why they're sitting in the as a common variant. But if you look at the rare variants, that variants probably the new variants coming out as a the de novo in a very near generation. So let's say probably up to 30 generations in the past, the variants appeared in the East Asian, and then the variants segregate to the following generation. So that variant only appeared in East Asian, or only appeared in European, or only appeared in other other populations as well. So rare variants is, that's why rare variants are only shared little portion across the population. And of course, if you look at the ultra rare variants or private variants, the level of this sharing proportion is probably much smaller than that percentage. Okay, and also the rare structure variation are also population specific. So if you when we compare the rare or common variants, we mostly use the SNP or index, because these are the easiest way we can screening the genotype information. And the later phase of the studies, I mean, the genomic studies normally following up with the structure variants, because the finding structure variants is much trickier than the SNP or index. So phase 3 of the 1000 Genome Project, they're looking at the structure variants, and they also compare whether these structure variants are population specific or not. They found it's population specific as SNP and index, and these structure variants are much rarer than other types of the variants. The reason is that the structure variance is large enough, and it should bring the largest impact than other types of the variants, right? Okay, so there was the implication from the 1000 Genome Project. And then we're looking at the what is the factors driving this population structure in our evolutionary process. And related to this, we know the 4 factors of the evolution. So this is the ideas proposed by Wright in 1940s days, 42 something. And 4 factors of the evolution is also referring the factors driving the population structure in humans as well. And also, of course, the variation in allele frequency throughout the time. So what are the 4 factors? So first one is the mutation, and the second one is the natural selection. Third one is the genetic drift, and the fourth one is the gene flow. And I guess these 4 factors idea is the last one you learned from the high school biology, and this is introduced as a Mendelian population or something. But anyway, that is proposed by Wright in about half centuries ago. And these 4 factors is the sort of the major topics when we discuss about the allele frequency variation and population stratification. Okay, so the first one is the mutation. So mutation here is a new DNA variant in the generation. So we called it the novel variant, right? And this is the ultimate source of the genetic variation. So new source of the variation coming out in the species, and it will have certain phenotype. It will have the new phenotype in our population. So if you think about the human mutation rate, this is about 0.0 to 0.5 times 10 to minus 8 per base pair per generation. So this is about 70 de novo variants per individual, so which is the 30 de novo variants with each gametes. That was the first estimation, and these numbers is way higher than nowadays. And if you calculate using this information, now we have about 60 to 75 de novo variants with each gametes, right? Okay, and what we know about the de novo variants, 80% of the variants from the father's sperm, and also, okay, so as I mentioned, that around 70 de novo variants from the parents. So if you compare your genome to your parents, you have the 70 different loci in your genome to your parents. And de novo variants may provide a new trait to an organism, right? So this can be work in humans, but also new mutation can be appeared in other types of the biological organisms as well. So let's say if you, I mean, the well-known examples in these days, of course, is COVID-19. So if you look at the news media, they always talking about the the new variant or new variation, and that is all characterized by the new mutation appeared in a COVID-19 virus, right? And because of this virus, we're sitting in the this cyberspace and meeting in as a cyberman. But anyway, so the novel mutation, if you're going through many generations, and certain generation which we have certain mutation in an organism, right? So this caterpillar has the mutation on this site, so which has to be G in this DNA, but there is the change to T somehow by the de novo mutation. And because of this de novo mutation, this caterpillar has the shinies in their in his or her trait. I don't know their the sex, but anyway, it became a Chinese caterpillar, and we call that this is the new mutation and new traits coming out in the in this organism, right? And then mutation work as a sort of source, but the actually the working mechanisms, whether that the new mutant sitting in the population or wiped out, is depends on the natural selection, right? So natural selection, of course, one individuals with certain genotypes are more likely than individuals with other genotype to survive and reproduce, and thus to pass on their alleles to the next generation. So natural selection originally proposed by Charles Darwin and talking about, I mean, which individuals is favored in a certain environment, right? And this is ultimately makes our makes us the variation in a population, and also the trait variance across the individuals. So we know about this variation is heritable, and also this variation is associated with the fitness and genotype, and all of this information, they interplay to shape our allele frequency in a population, right? So we're back to the new mutation and the caterpillar with the shinies, right? If the mutation is beneficial, then these individuals, they have much more numbers in offspring generation, and after a few generations, if you look at the population, the majority of the the population probably the shiniest caterpillar, right, compared to normal caterpillar. But if this mutation is harmful, that means that this mutant is not survived to certain individuals so easily, exposed to the bird like Pokemon, then this Chinese caterpillar will be removed out from the population. So after a few generations, we're not going to see the individuals with that trait, and we only see the normal caterpillar. And one of the measurements we represent that situation is the fitness. Fitness is the relative ability of genotypes to survive and reproduce. So if you're looking at the certain trait, and we can think about whether this trait is favored or unfavored in terms of the survival in the population. Okay, so people think about the many different selection models, but here I only put the two things. One is the directional selection. So in certain population, we have the direction of the favorance, right? So let's say if the dominant organisms are they favored in certain population, like the black moss in a certain whatever that the pollutant evolution, then the individuals with that genotypes is segregating in a population and sitting on the major the proportion in a population. But in other situation, then without the pollutant, then the light color moss, they're likely to be sitting in the population. So it is describing whether which trait is favored in a certain population, right? So if you're looking at how soon they will be occupied in the population of their genotype, then we can describe by whatever that dominant genotype or additive genotype or recessive genotype, right? So additive and dominant genotype soon be occupied in a population because they are working in a dominant situation, or additive is the only working in a one pair. So they easily saturate the allele frequency in a certain direction, but recessive is the 25 chance of the selection. So it takes some time, but ultimately after a certain generation that will saturate it in a population, right? So that is the selection situation with the directionality. But certain situation, we don't have the directionality. So in that case, the heterozygotes is way preferred in a population. So in certain situation, like in robins, when they take up the eggs by different hatch, they have to be sitting on the heterozygote situation. That is the way the better survival rates than dominant or recessive situation because of their the maintenance. So in that case, the heterozygous genotypes is the higher fitness than homozygous. So their allele frequency is always sitting in the across the 0.5% over the time. So there is no sort of the wave flavored way over the recessive or additive or dominant situation. Okay, so well-known examples of the heterozygote preference or the heterosis is the situation of the sickle cell anemia, right? So if you look at the sickle cell trait in an African population, we have to be the balance between resistant to malaria situation or susceptible to malaria situation. So if you have the normal the sickle cell type, then you're going to be quite fragile to malaria infection. But in other words, if you have the recessive genotype, which is the abnormal shape of your cell, it's definitely resistant to malaria, but also the fatal to the sickle cell disease situation. So if you look at the African population, so that's why the heterozygote carrier has much more high proportion in the population because of they have both the ends in terms of their genotype selection. So in this case, if you refer the heterozygous superior situation, the sickle cell anemia is one of the examples. Okay, and the third factors is the genetic drift. So genetic drift is the meaning is that how population behave over the time and how population is divided into the small parts and how they act in a certain environment. So as we learned in the co-lengthen theory, the populations in a natural environment, we work as a stochastic process as to their behaviors, right? So if you're looking at certain population and that population maintained as a whole in the population, but they can be divided into the small piece and they move on to the different area, right? So population with a small size can experience substantial change over time in terms of their gene frequency because of the random effect from the environment. So if you're looking at the small population and they migrate to the new place and they starting from the small size of their genetic backgrounds, so they are way beyond the random mating and they segregating the certain loci in their genome, and sometimes that is in favored in terms of the natural selection. And this genetic drift can reduce the genetic variation in our population. So people always try to measure the consequence of the genetic drift using the effective population size. So effective population size is the description of the size of an ideal population with the same rate of the genetic drift of gene frequency as the actual population. So if you think about the small population divided from the large population, then whether this population is effectively sitting in the certain environment and they can be survived over the time. So one examples of the genetic drift in our history as the Finland people. So I think I explained last time, the Finland people, they experience quite unique history of the subpopulation migration. So in their geographical area, lots of different the tribes, they migrate into the area and they settle down and they expand their population size over the long history. So early settlement area is the two colonization waves into the Finland, the suburb, in about 6,000 years ago, and they are segregating their population in the time, and they don't have really the big, the swap of the population, and they don't really share their the population with each other because different, the geographical regions and also linguistic region and also the cultural aspect as well, and also the religious issues as well. Because sorry, because the old people, they always met that situation. And also segregating at the area, and also due to the rapid expansion of the population, their population size is limited, and they are with the small genetic background, they are sitting in the time in a long ways. So the early people, which is the founder population, because of the small size of the genetic pool, they experience lots of different health condition issues and also disease, because they also segregate sort of the deleterious condition to the next generation, and also experience lots of the recessive situation in their population. And over the many generations, they didn't really notice out their issues because we don't have the modern science at the time. But I mean, over the half century, we now know about the genetic information and genomic technology, and people collecting the health condition over the many different areas, and they start looking at their own problem in a public health situation. So they're looking at all different types of the clinical conditions of the regions, and they can start to think about whether this, the found effect is, the impacting on their health condition because of the genetic drift consequence. And also another, the well-known examples of the consequence of genetic drift is the Amish population in Pennsylvania. So because of the religious issue, some of the Anabaptists migrate from the Central Europe to American continent, right? So Anabaptists, they including like Amish, Mennonite, or Wright. They're normally in the Central Europe, but because of the religious issue in 19th and 20th, they 18th and 19th, and they migrate from the Europe to the North America, and that was the sort of the big deal in a European country. But I think the religious issue is also the major force of the human behaviors, also migration in our history as well. Okay, so these Anabaptists, they also have quite unique the cultural differences. So they segregating the environment, they segregate isolated environment, and they only are married to themselves, and also I mean, they start from quite small number of the founder population. So of course, I mean, after a few generations, they experience the severe the clinical conditions, right, or the disease as well. So Victor McCusick was interested in that situation, and he started looking at their sort of the recessive patterns of the disease from their the genetic background, right? So he started to curate that information by himself. So I mean, at the first time, it was by his hand, right? So just the written record. But after the internet appeared, I mean, surprisingly, you never imagined the internet appeared as a sentence, but that time internet is the thing. So he curate all of this information into the online version, so called online Mendelian inheritance in man. So he curate all of these recessive condition and share that information with other clinicians over the world. And we gained the knowledge about the recessive disease and also the rare genetic information of our history, right? So I mean, the Amish population or these Mennonite or Hutterite, like Anabaptists, they are sort of quite interesting population as to the genetic drift and the clinical research. I mean, I only mentioning the two populations as the genetic drift. That is the well distinct population in our history. But I mean, the genetic drift is quite natural process. Many other populations also experience over the history, right? So I mean, people still doing their research in terms of how the human activities are impacting on genetic drift, right? So one interesting theme is like after Second War. We are start to be urbanized, right? So people start move to other place to study higher education. I mean, these things happen in Europe, America, or our country as well, right? So I mean, people start comparing the genetic information of non-migrate people to migrate people. So if you think about the people with the frequent migration to urban place, they are likely to be the higher education compared to the resident sitting in the certain area without the migration. They are likely to be in a rural industry or agriculture situation, and people start comparing the genetic information how these human activities is differ by people, and also how these things shape our certain traits or other phenotypic values as well. So well-known examples of the Korean situation is the longitudinal cohort of Ansong and Ansan, which is the Ansong is the well-known city of the rural industry, so people not really migrate and people dedicate to the agriculture industry. Ansan is the well-known city for the immigrants or the people from other other suburbs. So because this is the sort of the industry area, so people comparing their health condition and also genetic information as well. So that is the one example we can refer in our country. But also we are still doing sort of the similar effort on seeing more different examples of how we understand human migration and also health condition and how we understand sort of this genetic drift in our public health. So we are also middle of that progress. Okay, so gene flow is the also called homogenizer, which means that how we shuffle to each other. So I put all long description, but easiest way is that according to coalescent lineage, we are having different populations, right? And this population diverge to subpopulation. So let's say this population diverge to population 1 and 2, diverge to population X, population 3 and 4. But sometimes we also have the mixture of the between the populations, right? let's imagine that this population 2 and population X, they are quite, geographically close to enough. Then population 2 can be merged to population X, or population 3 and 4, they merge to population X, right? Population 2 and 3 and 4, they are merged in the place between these two area, right? So gene flow is how these populations are mixed to each other. So this is the examples from the butterfly from the South America, right? So from the Mid America, which is between the Mexico and Panama, right? And this is the upper part of the South America, right? And like Peru, or I think it's I don't know this area. Here is definitely Brazil. And how these butterflies, they are merging, and how they mix their population, and how these mixtures are making a sort of the new population in consecutive generations. Okay, and also, I mean, gene flow can be applied to human population as well. So I mean, I assume that the one of the interesting topic in, I don't know, maybe these days or your days after 5 or 10 years, probably one of the most interesting topics in human genomics is how we understand the interracial marriage population, right? So I mean, we covered the issue of how we define the disease genes from the genetic studies. I mean, that was covered in the session 5. But when we normally do the studies to look at the causal genes in certain disease, we looking at the same ancestry population. So that is the one way we design the experiments. So that case, most of the individuals with the mixtured ancestry are excluded, but I mean the proportion is quite small. So when we compare that genetic association, so I mean people normally compare the whatever the East Asian or Europeans or something. But in reality, I mean, more and more these days, we see more couples from the different cultural background, and also when we define when we study sort of genetic studies of the genetic research of the public health in our society, and we kind of define sort of Korean as a just ancestry information. The reason is that the constitution of the people in our society, we definitely have the major part of the Korean individuals with the Korean ancestry, right? But we also have the immigrant as well, and that those people also has to be considered as a sort of subject to the genetic research. So that's why we can think about the sort of the solution, how we create sort of how we approach the clinical interventions or developing the treatments to those people, right? So when we do research, I mean, not only the genetic research, but other biological studies, we are doing that because of the public, the marriage, right? And this is why government support our research. But that case, the genetic studies is always, and also usually, and also expectedly, supposed to use the sort of the same ancestry information for association studying. But if you looking at our the composition of the society, we now see more and more people from the multiracial background, and how incorporate those people into our study design. That is the sort of one sort of sort of agenda we have to deal with the in a future research as well. But and also this is also has to be considered in a clinical situations as well. So I probably explain again in session 5, but these days the research is mainly based on the the middle age the adult males, right? So the one simplest dimension missing in this situation is the female biology, right? And if you're looking at the the side effect of the vaccine, like an AstraZeneca vaccine, people talking about the vascular issue in young females, right? The reason, we don't know the exact mechanism, but the problem is that that situation also the development is also under the male biology, right? So one of the easiest dimension we can deal with the public health approach is to looking at the the different sex, right? So that is the way we can expand our knowledge into the public health. But the movement is already going on, because the sexual dimorphism is the one of the easiest way, and people already start to think about how we incorporate that issue into our public health, right? There was the 2016 and 17, and nothing really unique these days. But if someone asked me pick up the things we have to deal with in the future, the public health and biology, and I would pick the the international marriage or the multiracial situation in our population. So because we are facing that situation in our clinic, but also in our biological domains as well. So if you're interested in the genomic perspective or genomic medicine, then definitely understanding the ancestry is probably the one of the the urgent the agenda we can think about in our population in our research. Sorry. Okay, so let's move on to the models of the population structure. This is nothing really for questions for exams or tests. I'm not gonna make any test for this topic, because this is the mere introduction of the population models, because we are talking about how population behave in a natural environment. So I briefly introduced what kinds of models we are putting into over the history, right? So the easiest way of the population evolution is from the one lineage then separate into certain descendant or divergence, right? And divergence has the many different dynamic ways, or sometimes they merge to each other, and all of that situation has their own unique vocabulary to describe the situation. And we have the 4 major models to explain this population migration and divergence. So the first model and the easiest model is the island model. This is proposed by Wright, and this is one of the simplest way. So there was a large population, and there are several subpopulation migrate to each part, right? So this is like spreading out like a tiny island. And stepping stone model is the evolved way in each step. So migrate to here and to here, or to here, or sometimes they backward to the previous step. So this is like a the movement between different local region, right? And then isolation by distance model. So they have the major movement in their population by the different subpopulations. So they are in the geographically, they are in the largest chunk, and within the chunk, each population, they are migrate to different places. Or these types of the migration also happen locally by the small population. So we call the meta population. So from the A population, then the small population bud out from here, and they also migrate to other populations as well. So if you're looking at the population model, we don't have the one general rule to explain every situation. People still trying to explain the best model for the population migration, but I mean, still the field is debating. I mean, which model describe the best situation in a certain behavior and phenomena, but there has to be explained case by case. So I leave as a introduction here. But more important thing is to is the topic how we estimate this population structure. So there are a few ways we can estimate how population are different to each other. And one way is the fixation index, which is the first topic. And the second one is the PCA, which is another classical way we can looking at the population structure. And I'm going to introduce the examples of this population structure. Okay, so what is the fixation index? So fixation index, or FST, is the explain the situation how population diverse and how population differ by genetic structure. And this is the one way we measure that phenomena using the mathematical way. So that is originally proposed in 1951 by Wright again. And FST, as I mentioned, that this is a measure of the population differentiation due to the genetic structure. So if you're looking at the lineage, then we'll have the two different population. And if you're looking at these two different population and measuring the fixation index, and we can measure how much they are different to each other, right? So these two populations could be totally different. These two populations could be quite similar, or these two populations could be same, right? So fixation index is the quantify of how two different populations are similar or not, right? So how we measure the fixation index? We use the level of the heterozygosity, right? So if you're looking at all of the population and you measure the heterozygosity, which is the how many heterozygous variants in the population, or you can also measure the how many heterozygotes, which is the individual with heterozygous variants, and then you measure how many of them within a total population, which is all of the subpopulation. Then you also measure the average heterozygosity within each subpopulation, right? So here if you measure the HT, which is you measuring the heterozygosity from all of the population here. If you compare the African and European, then you measure these heterozygosity from all the individuals. Then you measure the average heterozygosity with each subpopulation. Then you can easily calculate the fixation index, which is the HT minus HS, and then this difference is divided by HT, right? So if you think about that, the heterozygosity differences is the same between these two populations, then you'll end up with the zero fixation index, right? So that case, all variation exists within a population and none exists between population, right? But if the fixation index equal to 1, then all variation exists between population. That means that two populations are totally different, right? So here has the examples. So if you look at two population, and I put this is the just the example. So you're not going to calculate the exact the coefficient using these numbers. So don't try that. So this is the every year question. This is just an example, right? So we have to use the allele frequency numbers given to each genotype. So I'm not giving you any genotype frequency in here, so there is no way you can calculate by then. This is just an example, right? Okay, so if you look at population 1 and 2, these two populations has totally different genotype in their population. So in that case, we can say that these two populations genotype information are totally different. So the difference is explained by total genotype information. So that is the fixation index is 1, right? Then if you look at the second scenario, which is the genotype compositions are same, so you can say that the differences between these two populations is nothing. So there is no difference. So there has to be 0. And all the difference is actually coming within the population, right? And if you're looking at this third scenario, we have the fixation index is 0.11. So which is the sum of the the differences coming within a population, but also between the population, right? So in that case, you can measure that the differences between these two populations. Okay, so if you look at the average fixation index between the population, you will see about 15% difference. So this is the numbers from the 1000 Genome Project. So if you think about the common variant situation, we have about 85% of the genetic variant is the shared by the population. So you can estimate that rest 50% would be explaining the differences between the population, right? So if you're looking at these fixation index and looking at the genomic research, then you can estimate how much the populations are different to each other. And this is the result from the 1000 Genome Project. So they use the synonymous variants from the 1000 Genome Project, and they measure the allele frequency of these synonymous variants, and they calculate the fixation index from the examples. So you might ask a few things. So first one is definitely, we are looking at the convariant definition or whatever, the rare variant definition, and also how the fixation index is working in a population. But then another question is why we use this synonymous variants, right? So the reason is that the synonymous variance is not subject to the natural selection. So this is not really changing any fitness in our population, right? Synonymous variance is the variance not going to change any amino acid in our population. So synonymous variance is represent, almostly represent the neutral variance in our population. But if you're looking at the non-synonymous variants or stop codon variants, these variants could be affecting the fitness or probably favored by environment, because they have different types of the protein, and the protein could be changing the fitness in a certain population, right? So let's imagine that Nepal people, they have a certain non-synonymous mutation, and the mutation is adapted to high altitude, right? So how could we compare that non-synonymous variants to other population, right? So this is very specific to a certain population. So here, why people use the synonymous variants, because synonymous variants not going to change any protein, and it's not going to affecting on the fitness. Okay, so also we can measure more specific fixation index across the certain area, which is the sub-Saharan region in Africa and also Middle East as well. And also they can compare their fixation index to each other too. Also, we can use more localized ways. So people can looking at all different European countries and European ancestry and looking at whether there is any population structure in a European people too, right? So this is the one way they can do that. Or another way we can measure the differences is using the distance. So distance is the differences between the allele frequency, differences of allele frequency between population, okay. So distance is the, the simple way to, measure the differences in any mathematical situation. So if you think about the genetics, you can utilize the allele frequency information, then you can measure the differences between the population and one and two, right? So if you're using the SNP1, then you can measure the differences between the population 1 and 2, 1 and 3, or you can use the SNP2, you can measure the differences between population 1 and 3. Or you can measure the differences across the SNPs and you can average out their differences between population, right? So if you think about this distance, then you can easily think about the way between the coalescence situation. So from the founder population, you can describe the first the population diverge from the origin, and then you can also think about the second divergence, right? So if you think about the out of Africa situation, then you can think about the African population versus non-African population. Then you can think about the two population diverged more recently, which could be, East Asian versus, South Asian, something. Okay, so if you infer the population structure from genetic information, then the easiest way we can use is the factor analysis. So one of the well-known examples of factor analysis is the PCA. So PCA is also sort of the source of the clustering analysis. So if you're looking at the individuals for a certain population, then you can make a cluster between the different population. Then here you can also use the PCA information to do that, right? Okay, so what is the factor analysis or the PCA? So factor analysis is a class of the procedure used for data reduction and summarization. So factor analysis can be used to identify underlying dimension or factors that explain the correlation among sets of variables. So, and also factor analysis identify new smaller sets of uncorrelated variables to replace the original sets of the correlated variables too. So if you're looking at the certain data set, if you want to find sort of the direction, or if you find some certain dimension to explain the differences between individuals, in that case you use the factor analysis, right? So one of the examples of the factor analysis is the PCA, which is the principal component analysis. So what I meant, the data set, which is the genotype here. So genotype means that genotype data means the matrix, so which is the column is the number of samples and the row is the number of genotypes. So if you're looking at the genotype data set, you have the samples which are individuals from that project. So if you think about the 1,000 Genome Project, there are 1,000 participants in the data set, which is the samples here. Then the row is the number of genotypes. So we measure the genotypes from the individual, right? So this genotype data set is combination of individual ancestry proportion times subpopulation allele frequency. So if you're looking at these people's genotype information is the mix of their allele frequency and also their underlying ancestry information. So this is the mixture of different informations of the data set. So we have to looking at the dimension to explain this mixture, right? So first thing you can do that is you decompose this data set into the several dimensions. So that means that if you have the genotype information, then you have certain vectors to explain that dimension. Then in the end, you will have the individuals in the plots, right, like in here. And then you can explain that these individuals from the certain population, let's say population 1, population 2 and population 3, because we know that the allele frequency is explaining the population structure, right? So this is the one assumption. And then this underlying population structure has to be represented as a differences in an allele frequency, right? So if you're looking at the allele frequency, it has to be decomposed, which means that it has to be divided into the, not divided by, sorry. It has to be reduced into the, into the ancestry information, right? So if you look at the genotype information, that data set has to be explained by different population, right? So what PCA does is that they're looking at the first dimension of explaining the largest variance of the data set. So PCA looking at different dimension, here has the principal component. So from the principal component 1, 2, 3, 4, and certain, and nth, right? So it can be up to, you know, hundreds, thousands, or millions. It's up to your operation. But normally, the first principal component explains the largest variance of the data set. So data set here means the genotype, frequency, allele frequency, right? So what is the major, you know, the factors explain the largest variance in the allele frequency of the multiple individuals? That is the population structure, right? So if you're looking at the data set, then it has to be combining into the, it has to be reduced into the certain individuals explained by different populations, right? So if you're looking at certain dimensions, so from the first dimension, second dimension, and third dimension, then it all indicating the differences coming from the population structure, right? So this is one example from the migration history in Eurasia. So if you're looking at the HeMe population data set of the and then HeMe population plus in Indian population, then they can well separate by their population within a PCA plot. So this is the PCA plot, PC1 and 2. Then you can see each individuals, here is the single dots. They are clustered by certain populations. So Thai people, they cluster each other. Cambodian, they cluster each other. Vietnamese, they cluster each other. Japanese cluster each other. And Pakistani or whatever, the Nepal people, Kyrgyzstan, or the European people, they cluster each other because the population differences is explaining the largest variance in a PCA. So if you're looking at PC1 and 2, that has to be well represented in a PCA situation. And this is the examples from UK Biobank project, and they looking at the hundreds and thousands you know, England people. They collected the people from the UK society, but of course some people from the native, white British. This is what they called in their study, and also white Irish. I think this is quite interesting nomination, but anyway, this is the what, you know, they explained from their study. But also some people from South Asia or East Asia also in the society, so they are present in the PCA plot. But here is the different ways. So this plot is the PCA, so PC1 on x-axis and PC2 is the y-axis. But if you're looking at the population structure, it's explained by PC1 and 2, but PC3, 4, 5, 6, 7, or sometimes PC20 or PC30, they're all explaining the population structure because the population structure is quite obvious driving factor for the differences of the allele frequency. So more recent technology from the machine learning, they merge these different pieces. So here, and they represent the stratification. So they using the latest technology called UMAP, and this is the different, you know, representation to PCA. So what they done is they merge PC1 to PC10. So that's why they say first PC10 principal component, right? And they merge together and they put it into the two-dimensional space, right? So if you merge 10 principal component, that has to be 10 dimensions. 10 dimensions, you know, human cannot read it. So UMAP is the technology you merge multiple dimension into the two-dimensional space, then you can looking at their structure. And this is also, you can see the population stratification in NOMAD project. So this is again the human differences you can measure from the allele frequency. So if you're looking at the PCA for the ancestry inference, you can easily detect the direction of the major variance in our data set, right? So you can easily see the population differences between subpopulation, and also this is also using for the association test. We'll cover this in session 5, not 4. Okay. And the last one is, if you, I mean, I explained last time, but if you read this population information, you always care about the differences between the race, ancestry, and ethnicity, because this is always working in a different situation and also different terms as well. So you can read some, I mean, great article from the David Wright or Francis Collins. They discuss about how this, you know, the terms has to be appreciated in our research, and also a fun video about the, you know, the mocking of the whatever the racism issue in genetics, and they talking about the supreme genetics parody. I mean, you will find fun a lot. Okay, so summary the topic. So we talked about the how allele frequency used to describe the population structure and also the factors of the evolution process and population diversity. And also we are learning the how to measure these populations stratification using fixation index and PCA. And as usual, we have a bit of the further readings for the population structure. And also, if you're not really familiar with the mathematical notation about the factor analysis or the PCA stuff, I mean, this is really 101 things. This is the really basic things we have to learn in the learning in science. So, I mean, I hope the basic math class in first year covered this issue, but I mean, unlikely and unfortunately not. So if you're not really familiar with this, so going to this link and think about, and also always welcome to leave your question on the Slack channel about, you know, topics we learned in the session. Okay, so I think time's up, a little bit past. So we continue to inbreeding, just inbreeding next Wednesday. And okay, see you next time.